такий, знаєш, пласт, десь може він тут лежить, ти зможеш побачиш.
We continue our last day and last presentation by Danny Duran. We have interesting lecture on what is it? A horizontal transfer gen. So Danny Duran, Carnegie Mellon University, USA.
Thank you. So can everybody hear me online?
Yes.
Or I'll keep right on going. So I'm going to I chose to tell you about this particular project, partly because it's one of my favorite projects, but also because it illustrates the importance of Theodore Jablonski's vision and why molecular biologists and cell biologists and people who are interested in fundamental medical applications need to know about evolutionary biology and how to interpret trees. So next, oh. I'm not advancing here. Is there a, aha, I am advancing. So I got involved in this project. There was the project sort of started, or the first part of it, a graduate student at Princeton University characterized an enzyme in yeast, which was not previously understood, that provides a different pathway to synthesis of ribose from the two canonical pathways that were well studied. So these are the two canonical pathways. That's not what I had in mind. Which button is my my mouse, my pointer? Aha, okay, these are the two canonical pathways. And on the top, you see the oxidative pentose phosphate pathway, where ribose is the result of a series of reactions that start with glucose 6-phosphate. You can also find in most eukaryotic cells the non-oxidative pentose phosphate pathway, where in this case, the starting point is fructose 6-phosphate. So it's a step, several steps down in glycolysis, and it works its way up back to ribose. So here's a cartoon of the two pathways I just showed you. You have, whoops, you have glycolysis at the top. So here's glucose, and it works its way down to pyruvate. And there are two places where this branches off to make ribose, which is essential for catalytic RNAs, for regular RNAs, for deoxyribose nucleic acid, so fundamental to life. And these two were very well studied until Michel Klaskin's, and it was thought that we knew everything about central metabolism in yeast. It had been the target of study for 50 years until Michel Klaskin's PhD thesis. So this is 2007. And at that time, about 15% of yeast genes were uncharacterized. Nobody knew what they did. And there was a project at Princeton to choose high quality targets for characterization, specifically focusing on things which were probably enzymes. And they did this by mass spec screening. So basically the idea is, supposing you have a series of reactions like that so you've got enzymes A, B, enzyme BC, enzyme CD, and so forth. And these are the metabolites that the intermediate steps in this reaction. So if you knock one of these out, you would expect to see a buildup of B, say you knock out this enzyme, and absence of C. So you could then screen for changes in metabolites using mass spec and look for cases that were promising for characterization of the enzyme as a whole. So, and there was one in particular which at the time was called YKRO43C. If you knock out YKO43C, you suddenly see this huge red peak here, whereas in the wild type, you had this little blue peak. And so Michelle took on the challenge of determining what it does, which reaction it catalyzes, what are the pathways it connects to, and so forth. So she determined that this was acetoheptylase bisphosphatase. So here, here's acetoheptylase bisphosphate, and what the enzyme she characterized does is it removes this phosphate group. And that provides a third pathway to ribose in the cell. So here you've got two intermediates from glycolysis. It creates acetoheptylase 1,7 phosphate, and then SHIB17, which is what she named this protein, removes one phosphate. We get to acetoheptylase 7 phosphate, and then back into the non-oxidative pentose phosphate pathway. And she also showed that, so this is a way of synthesizing ribose without changing the redox balance in the cell. She had indirect evidence that SHIB17 allows for rapid ribose production in low oxygen environments. And so this changed how we understand ribose production in fungi. And in 2011, this nice paper came out. So Michelle was understandably very excited about this. And I should say that she worked with a woman who then was a postdoc and now is a tenured professor, Amy Cotty, who's also shown on this slide. So Michelle was very excited about this, and she thought, well, okay, so here we have something that allows for rapid growth in low oxygen conditions. And what are important low oxygen conditions? Well, tumors might be low oxygen conditions. And, maybe I've got something here that would lead to a cure to cancer. And so she did some blast searches, and she found a protein in the human genome, and she started doing experiments with it, and none of her experiments worked. And finally, in consternation, I met Michelle because I was doing a sabbatical at Princeton. I had already gone back to CMU, but the phone rings one night and it's Michelle saying, do you think I'm doing something wrong with BLAST? Maybe I don't have the right human ortholog? So I went and took a look. And so the first question is, Michelle's assumption was that an ortholog, there is a human ortholog of yeast, and that's a gene that does the same thing that the yeast gene does. Is that the correct understanding of the word ortholog? No, it's not. What should it be? So an ortholog, two genes are orthologs if they share common ancestry by speciation. In other words, the branch point in the tree that leads to both of them is a speciation event as opposed to, say, a gene duplication. It has nothing to do with the function of the genes. Now, it's frequently the case that genes that are orthologs perform similar or analogous functions in two different species, but that's nothing about the definition. It's also, back then and probably still now, you could find many yeast biologists who thought that, an ortholog was the gene that in human that did the same thing that their gene did in yeast. And there's certainly not the case that every yeast gene has a human ortholog. So I went and took a look. Here's why Michelle ran into trouble. So SHIB17 is a member of a very large family of acidic phosphatases. And these phosphatases, there's a key histidine here that takes the phosphate off the substrate. And there are many, many, many different kinds of proteins that use this domain that performs the phosphatase function. And so this is from a review article, and it shows just a few of the different cases you have. And you can see that they have different multi-domain organization. And even if you just look at that phosphatase domain, they're so diverged that many of them you can't find by doing a sequence search. So you can think of this cartoon as showing the entire family of proteins that have this phosphatase domain. And then within it, you have different subfamilies that actually correspond to specific functions. And so here, this darker purple circle is supposed to correspond to the set that are actually orthologous to SHIB17. And SHIB17 has the domain. It has a tail of about 80 amino acids as well. This is an alignment that shows SHIB17 and its close relatives, and the red columns are columns that are specifically conserved in other SHIB17-like proteins. The yellow columns are ones that are conserved in other members, more distant members of the family as well. And so this gives us a signature of specific sites that we can use to try to find this subfamily and not any old acid phosphatase. And the signature, I mean, these key positions, you can see that they're important in the active site. And in addition, some of them are in this 80 amino acid long tail. Now, if you do a standard search with BLAST, it's going to pull out anything that matches this with or without the tail. So there's a tool called SciBlast where you sort of build up a more and more detailed model of the sequence you're looking for. There are other things that will do this. You can model it as a hidden Markov model, for example. And so I use that specifically to find this subfamily. And this is the taxonomic distribution of the subfamily. So you find it, throughout what's called the higher fungi. So these are the Basidiomycetes and the Ascomycetes. Not every species, but it's well distributed across both of those groups. It's not found in earlier branching fungi. It's not found in animals. It's not found in plants. It's not found in protists or archaea, but it is found in some groups of bacteria. And the largest group of bacteria are the rhizobia. So this is species that form nodules on plant roots and have a symbiotic relationship with plants in which they do nitrogen fixation. There's another group that does nitrogen fixation in the actinomycetes, including Frankia, and that has it. And then there were sort of isolated genomes isolated in the tree, associated with their soil genomes, bacteria that live in the soil, and they might, be found near plant roots, but they aren't nitrogen fixing. So this was interesting, and this explained why Michel's experiments in human don't work. There is no human ortholog of SHIB17 in any animal or plants. So how did we get this distribution where we have two distantly related taxonomic groups that both have copies of this gene? Well, one possibility is that you had some ancestral acid phosphatase that duplicated one copy specialized to perform cytoheptylase bisphosphatase function. And that copy was, according to this hypothesis, it was retained in fungi and retained in some bacteria and lost in plants and animals, and also from all the protist species that I haven't shown. So lots and lots of loss required to explain this. If this is the correct hypothesis, we would expect to see a gene tree that looks like this, where at the root, you've got other acid phosphatases on one side and SHIB17, SHIB17 on the other side of the root. Here's a second hypothesis. You have acid phosphatases evolving since the beginning of time. And then somewhere in bacteria, you have a duplication event that gives rise to a subfamily that becomes cytoheptulose bisphosphatase. And then following that, you've got a transfer into fungi. So now if that's the correct hypothesis, you'd expect to see a tree like this where you've got the cytoheptulose bisphosphatases nested deep inside the tree of acid, of the larger tree of acid phosphatases. And then the fungal ones would be nested inside the bacterial sequences. You could also have a scenario where they first arose in fungi and then were transferred into bacteria. And if that's the case, then you would expect to see, again, you see cytoheptulose bisphosphatase nested in the acid phosphatases generally, but the bacterial ones are within the fungal ones. So we've got these three hypotheses for the origin of this family. We can build a gene tree and then look and see, do we see one of these particular gene, tree shapes? So here's the tree. This is a selected set of species that have it, not all of it, or a selected set of cytoheptulose bisphosphatases taken from some of the species that have it. So here, these guys are the bacterial ones. You've got Agrobacterium and Rhizobia, you've got Frankia. This thing here is other acid phosphatases, which I used essentially as an outgroup. And then you see the fungal ones here. So we have fungal nested within bacterial, and those are then nested within other acid phosphatases, and that's consistent with a history where it first arose in bacteria and was then horizontally transferred to fungi. And I want to point out, we didn't quite get to this for those of you who were in the workshop, but for species trees, you can root a species tree by finding a species that's distantly related to the species in your in-group and using that to find the oldest point in your tree. Now, that doesn't always work with gene families, because it could be that there's no sort of natural selection of a member of the gene family in a species, so that's an outgroup that's appropriate. So in this case, I rooted this essentially with a duplication, and that's a handy thing to do, to use some distantly related gene family to root. Okay, so we have support for the hypothesis that fungal cytoheptulose bisphosphatases were acquired through horizontal transfer from bacteria. Well, how plausible is this as a hypothesis? Because these are distantly related species. Well, we note that many of the bacteria that have this are plant symbionts. And in fact, not only are they plant symbionts, but they have an ecosystem in which they meet. So that here's the nodules that would have the bacteria in this cartoon that perform nitrogen fixation. So there's an ecosystem in which you have close proximity. And then in addition, some of these plant symbionts actually transform the nuclear DNA of their host. So this is a cartoon version of it, that you have this plasmid with something called tDNA, which actually enters the plant cell and goes to the nucleus and gets integrated. And this is what happens with Agrobacterium tumefaciens, which is actually, it's a pathogen. It's not a symbiont. It's sort of, it's given up on its half of the deal where it provides nitrogen in exchange for nutrients, and now it just goes in and manipulates the plant cell to its advantage. So it's been shown that Agrobacterium, as part of its infection process, will transform the nuclear DNA of the host, but that it can also transform fungi if fungi and bacteria are found in the same plant wound. There's the plant excretes a hormone that triggers this infection process, and if there's some other fungal cell sitting there, it can also get transformed. So we don't know whether or not this particular transformation function was actually present in fungi at the time that this horizontal transfer took place, but it's sort of suggestive that, maybe there's actually an ecosystem and a mechanism which would explain how such a transfer could occur. Okay, so orthologous genes do not, by definition, the definition of orthology has nothing to do with function. However, if you have a putative ortholog and it also performs the same function, that's some evidence that, in fact, your putative ortholog really does share common ancestry and hasn't changed that much. So we tried to see whether or not the bacterial proteins could replace the fungal proteins for function. So this is from Michelle's paper. And one of the experiments she did was that she knocked out one gene in each of the 3 pathways to ribose and showed that when you do that, yeast has a dramatic growth defect. So this is a growth curve where this is time, and that's the number of yeast cells in a culture. And if you knock out all 3 of these genes, you can see that it's growing much more slowly. That's the red line. If you replace, you put SHIB17 back in the cell on a plasmid. So you've taken out the native one, and then you put it on a plasmid, you put the plasmid in the cell and grow with constitutive SHIB17, you get function back. So that's, I don't know if she's showing that here, but that must be, I don't know if she's showing that here, but when you express it constitutively, you do get it back. So we thought, well, we can try that with the bacterial gene. So we had all 3 of these pathways to ribose are knocked out in this strain. And then we're expressing either the native yeast SHIB17 from a plasmid. We've got the empty plasmid as a control. We've got the agrobacterium SHIB17. And then finally, we put in the closest human homolog. So this is an acid phosphatase, but it doesn't have those key positions that are characteristic of the SHIB17 family. And here are the growth curves. So the purple SHIB17 is red. SHIB17 from agrobacterium is red. SHIB17 from yeast is purple. And you can see they're growing much faster than the empty plasmid or the human version, which we, based on its sequence, suggests it's not an ortholog, and therefore there's less reason to expect that it would have the same function. So we've got this divergence event that presumably goes back at least to the beginning of fungi. So the transfer to the higher fungi. The transfer occurred before the separation of basidiomycetes and ascomycetes. So it's an old transfer, and yet the gene from agrobacterium can replace the function when we put it into yeast. So we've got information about which species have this pathway. We've got information that suggests the mechanism by which it could have been acquired by horizontal transfer. Usually if you have a horizontal transfer, it gets retained if it has some fitness benefit to the new host. So what could that fitness benefit be? So this illustrates various fungal lifestyles. These are photographs from Candida albicans, which is moderately close, depending on your viewpoint, it's moderately closely related to Saccharomyces cerevisiae, to baker's yeast, and it's one of the model organisms for biofilm formation in fungi. So you can see here, you're seeing regular yeast cells, you're seeing filamentous growth, and you're seeing a biofilm. Well, we know that there's indirect evidence that suggests that SHIB17 allows Saccharomyces cerevisiae to grow quickly when it's in low oxygen environments. And in fact, if we think about properties of biofilms or Candida albicans biofilms, low oxygen, from expression studies in Canada, we know that this particular gene is upregulated and the ribosome biogenesis is upregulated in biofilms. And then finally, we also see more glycolysis in process. So the process that breaks down glucose and provides the starting point for ribosynthesis using SHIB17. So that suggests, well, maybe SHIB17 contributes to biofilm formation. And so this is from my colleague, Aaron Mitchell's lab, and an undergraduate working with me, Leon Kim, and a graduate student of Aaron's, Jigar Desai, knocked out, and Candida albicans is that it's got two copies, it's got two copies of every chromosome, so it has to be a double knockout in order to actually see the effect. They knocked it out, and they looked at biofilm dry weight. So you're seeing, these are the biofilms, wild type biofilm versus the deletion strain biofilm. And if you look at the dry weight of dried biofilms, you see there's a modest reduction in dry weight when you take SHIB17 out. So Jigar looked at these under the microscope, and he looked at them some more, and he looked at them some more, and eventually he began to think that maybe these hyphae, these filamentous forms that Candida albicans makes during infection, a little thinner to him. So here you're seeing, you're seeing the wild type, you're seeing this is when you knock out SHIB17, and this is when you reinsert SHIB17 into the cell to double check that your removal of SHIB17 really is responsible for the effect you're seeing. So he did an experiment where he took slices of this thing, and he found the filaments, and you can measure how wide they are by squinting into a microscope. And he did that for a long, long time and got a lot of data. And what you're seeing here is in biofilms, hyphal diameter in biofilms, this is the wild type. These are two different replicates where SHIB17 was deleted. Here's what happens when you put SHIB17 back in, and this is a second double deletion. There's strain there's a paralog of SHIB17 and Candida albicans, so that we knock that out as well. And what you're seeing is if you don't have SHIB17, so these two and those two, you have, in fact, thinner hyphal filaments. So that's consistent with our observation that the biofilms are not as healthy, and also that this is the morphology that Candida albicans uses for invasive growth. If you do the same experiment growing Candida albicans in planktonic culture, just in liquid culture, you don't see this effect. So you'll see that here the knockout, the complement, the double knockout, these all have roughly the same hyphal diameter. So this specifically seems to be an effect, these skinny hyphae, that you only see in biofilm growth. And then this is the kicker. If you knock out the SHIB17, but you feed the culture ribose, you provide it ribose directly in the culture, it rescues the effect. So then this is almost the same diameter. It's not statistically different from either the wild type or the complement. So this suggests SHIB17 is important for hyphal formation, it's important for biofilm formation, and it's specifically linked to ribosome biosynthesis. Right. We see a modest reduction in biofilm mass. The hyphal diameter is reduced relative to wild type in biofilms, but not when grown in liquid culture. And the defect is rescued if you provide ribose as a carbon source. So all of this provides indirect evidence that suggests maybe SHIB17 is important to biofilm formation and invasive growth in yeast. And that's nice because it suggests a possible reason, not just do we have a possible mechanism for this horizontal transfer, but we have a possible reason why it might have been advantageous initially at that transfer, and it might have helped fungi to develop this new lifestyle, which is in some, there are industrial applications in which it's helpful, and then there are also medical situations where it's a killer. So what have I told you? SHIB17 drives the thermodynamically committed step in a novel ribose pathway discovered by Michelle Klaskin in 2011. Fungi likely acquired this by horizontal gene transfer. A history of ecological relationships between bacteria and fungi on plant roots suggests an environment that's suitable for such a horizontal transfer. In addition, Agrobacterium, which is one of the bacteria that carries SHIB17, in fact, it's the one that's closest in the tree, and it's the one we showed was able to rescue the function. So it has this ability to transform the nuclear DNA of other organisms, which suggests a mechanism, and further, the SHIB17 gene from Agrobacterium can function within yeast cells. So there, it has, in some contexts, it has the same enzymatic function. SHIB17 expression patterns in yeast suggest that it may support growth in low oxygen conditions. Low oxygen conditions are consistent with biofilms and with invasive growth, and therefore have the ability to grow in low oxygen conditions could be related to the development of this infectious lifestyle. And further, when we take the SHIB17 ortholog out of Candida albicans, we see both a loss in biofilm mass, and we see a reduction in the morphology. We see basically a morphological defect in hyphae. So these are the people who contributed to this project. Leon Kim was an undergraduate at CMU at the time. Amy Coddy, the postdoc from Princeton, who went on to get a faculty position at CMU. My colleague, Alan Mitchell, who's now at the University of Georgia, and his team, and particularly his graduate student, Jigar Desai. Ridham and Garg we did some follow-up studies to make sure that the growth curves were repeatable, and he was a graduate student at the University of Texas at the time. And it was funded by the National Science Foundation, the Human Frontier Science Program, and a family, the Berkman family in Pittsburgh, who comes up with small grants for people who want to try risky things. And finally, I wanted to make a pitch for Pittsburgh as a city. Some of you might be thinking about graduate school or postdocs, and we do biological sciences at Carnegie Mellon. We also, our neighboring, right across the street is the University of Pittsburgh. They also have a lot of evolution in their biological sciences department. The two universities together run a computational biology PhD program. And you heard from Taras yesterday about the importance of evolutionary medicine. And in fact, there is an evolutionary medicine center at the University of Pittsburgh, and people from other universities in Pittsburgh participate in that as well. Questions?
Thank you so much. So first of all, I was really thrilled to hear that there are like suggestions that probably we can have this sort of way of ribosome expression or like creating because of this gene. And basically in cancer cells, we have Warburg effect. It's like the way when cancer cells can ignore the usual ways and they can just do energy receiving without any problems. And my basic question related to the theoretical experiment, I mean, can we just try to use transgenic models and try to like create mice as probably with this gene to see do this works for mammals, and this way to understand maybe like we can use this for like maybe target therapy in cancers?
That's interesting. Having discovered that it's not in human cells, I stopped thinking about what we could do with it in human cells. But maybe there's a gene therapy possibility in the future where that could work. So maybe you want to do that next week, try it out. Make those transgenic cells.
Could you please repeat one more time? I couldn't.
Would you like to, next week, you could try that.
Yeah. I
Yeah.
would be thrilled.
Yeah. So yeah, no, but we did look to see if there was evidence that, you know, if you feed mice Candida albicans with this knocked out, do they have a lower risk of getting, you know, a fungal disease? And those experiments were inconclusive, but we didn't look at it as a possible agent for gene therapy like you're suggesting. It's an interesting idea.
Thank you for answer.
You show, thank you for your presentation. It was great. And you show slide when your team or your student knock out genes and they put the plasmid in to, yeah.
This one or the one in the earlier one?
Previous way put knock out genes and then students put the plasmid in to, yeah.
This
that,
one.
that one, yeah. So no, previous, sorry. Yes, this one. Is there any difference if we just knock out first two genes but not the
Yes.
SGB17?
Yeah, so let me be clear. This work was done by Michelle Klaskowin before I got involved. So she's not my student. But what you're seeing here is all the cases where they knocked out one gene or two of the three genes, and those blue lines are on top of the wild type line. And they didn't, as far as I know, they did experiments with nutrients. But in this experiment, I think they were growing it with normal, rich medium, not, you know, experimenting with different kinds of challenging carbon. So you might see if you lost one of those that you didn't get the same growth rate under some sort of challenging conditions, and they grew them under normal oxygen conditions. But in those conditions, you had to knock out all three to get the growth-free defect. Great question.
Thank you. Just curious, maybe genome is available and you can guess what the percentage of horizontally transferred genes in entire genome of this fungus?
Could I guess right now or? I can't, I don't have a numerical answer at my fingertips, but it's occurred. So this was an uncharacterized gene.
Okay.
And it's occurred to me that the uncharacterized, the ones that are growing by vertical descent and they're in, you know, mouse and human and model organisms that are heavily studied, probably, you know, those genes are easy to study from in terms of, is there a challenge with their evolutionary history? They might be difficult for other reasons. So I think the ones that may turn out to be transferred will turn out to be the ones that are understudied. Because, for example, they're not in human. And I've thought about, and maybe I should think about it again, one could take that set of uncharacterized genes and test them systematically for horizontal transfer. And then, you know, look at, you know, some program for seeing what, because they're not in human, they would be great targets for drug therapy because they wouldn't cause side effects, or they have lower risk of causing side effects. So I think that's a very promising direction.
Thank you.
I'll do that next week.
Is anyone question? No?
Is there any question from online?
No, no one. Oh, sorry. Yes, one question. Just a minute. Question from Valeria Vasilyeva. What can prevent horizontal transferred genes from pseudogenization?
From transfer genes from cell, can you repeat it?
What can prevent horizontal transferred genes from pseudogenization? Pseudogenization. I don't know. Pseudogenization.
Pseudonization. Well, I suspect there are probably many cases where they are pseudogenized because they're either deleterious or they're neutral. But we don't see those. We see the ones, if we see them, if it's an old transfer and we see it today, then somehow there was some mechanism that forced the cell not to get rid of the gene. So, in this case, we have an example where we have a well-supported hypothesis that it in fact was advantageous because it supported growth in low oxygen and maybe invasive growth. So probably many horizontally transferred genes either kill the recipient or are pseudogenized. And we just don't see evidence of those unless it was, transfer that happened, say, last week. So thank you and good question.
Thank you. Yeah. Thank you.
And I'd like to say, I've had so much fun at this workshop. So thank you for inviting me and thank you all for providing great discussions.
Rocky. Come here.
So we finished our session all, and we finished our program for all days of First Ukrainian Evolutionary School of Evolutionary Biology. But now, unofficial, I would like to come up on the stage for common picture. Окей? Будь ласка, всіх запрошую піднятись на сцену для спільного фото. Фото. На завершення я би ще хотів надати слово проректору з наукової роботи Ужгородського університету. Да. Це не кінець. У нас ще трошки офіційна частина триває. Іван Святославович Миронюк.
Доброго дня, шановні учасники української школи з еволюційної біології. Мені тут дали таку шпаргалку, щоб я все правильно сказав. Хоча я додам багато що від себе, свою думку і бачення. Ну, перше, я хочу вам подякувати за активну участь у цій п'ятиденній школі, яку організував наш університет, Ужгородський національний університет, спільно з Харківським національним університетом імені Каразіна. За фінансової підтримки Європейського товариства еволюційної біології. І ця школа об'єднала талановитих учених, дослідників, практиків з різних куточок світу. США, Франція, Японія, Нідерланди, Румунія, Велика Британія, Польща, Ірландія і Україна. Я вам скажу своє враження. Не враження від школи, бо я на ній не був присутній. Більше скажу враження від, скажімо, моя думка, коли я побачив анонс, що така школа буде проводитись в Ужгородському університеті. Школа еволюційної біології. Ну, перша школа. Я дуже радий, що такий формат, як школа, почав відновлюватись в Україні. Ми проводимо в нашому університеті величезну кількість науково-практичних конференцій. Ну, я думаю, що буду дуже близький до істини, якщо скажу, що щотижня в Ужгородському університеті проходить якась науково-практична конференція. Це може бути всеукраїнська, може бути з міжнародною участю, може бути студентська. Конференцій дуже багато. А от шкіл досить мало. Хоча, на мою думку, на відміну від науково-практичної конференції, яка є більше майданчиком обміну досвіду між науковцями, школа є набагато більш, на мою думку, ефективним інструментом передачі знань, ніж конференції. Чому? Тому що на школі виступають дослідники досвідчені, які набули нові інноваційні знання і передають їх з перших уст молодим майбутнім науковцям або молодим науковцям. І ефективність такого формату набагато вища, ніж любої науково-практичної конференції, на мою думку. Якщо брати одну із базових статутних зобов'язань і функцій закладу вищої освіти, коли мова йде про освіту. Тому то, що українська школа пройшла в Ужну, я вас з цим вітаю, це чудове надбання. Друге, еволюційна біологія. Скажу щиро, я не фахівець, не у біології, не тим більше в еволюційній біології, але знаючи, що це таке, я вам скажу, що я є представником галузі знань, ну, навіть галузі практичної діяльності, галузі наукових досліджень, яка є основним реципієнтом та навіть бенефіціаром тих результатів, які ви здобуваєте в еволюційній біології. Тому що саме медицина, саме громадське здоров'я є тими сферами, де практично впроваджуються результати досліджень в сфері еволюційної біології. І, по-моєму, самі проривні винаходи, нові методи лікування, нові методи діагностики, в сфері медицини і в сфері збереження здоров'я населення були побудовані на досягненнях, в тому числі і еволюційної біології. Тому я вас щиро вітаю з цим чудовим заходом, який був проведений у нас в Ужгородському університеті. І бачачи багато молодих облич перед собою, хочу вам побажати значних успіхів і досягнень в сфері вашої і практичної діяльності. І для мене, як проректора з наукової роботи університету, важливо науково-дослідної діяльності, якою ви займаєтесь. Я дуже сподіваюся, що будете займатися, і, можливо, колись хтось з вас, я в це вірю, стане Нобелівським лауреатом в сфері біології, чи в галузі біології, і згадаю у своїх мемуарах, що приймав участь в Українській школі еволюційної біології. Вітаю вас і бажаю вам успіхів в усьому. І слава Україні!
Іван Станіславович. Ще декілька слів від імені організаторів. Наталія Волкова. Так? Буде презентація? Якась коротка.
We are going to make some concluding remarks about general experience that we have got here. First, Alexander Maestrenko will represent. Alexander Maestrenko will represent our statistics, if it is possible to say in such a way. In parallel, I will send to our WhatsApp group the survey for participants to answer some questions for our further evolution of this event. Alexander here? Yes.
Maybe you cannot hear me still.
Here, we hear.
Okay, perfect. Okay, sorry. Yeah. All right. Yeah, this is very improvised. Sorry. This is very improvised summary of what's been done, what we think will happen next. And yeah, again, it's nice to organize this event, and at the same time it's very sad now to understand it's been almost complete. So with this, yeah, a couple of words of what has been done and how it will go next. So first of all, we would like to thank the European Society for Evolutionary Biology, who provided support to organize this event. We're also very grateful to all other co-organizing institutions who really enable this to be possible. And yeah, I think, so this photo is, I believe, from opening of the school. So we really hope you had great time, made new friends, new connections, maybe for future collaborations, and to have something to look forward and something to remember that it's been a great event. Yeah, so this is us, the organizing committee. So we roughly started in July or August, I think, meeting first times to actually plan it. And so we are scientists, so we are of course interested in numbers. So how the event went, numbers of applicants, etc. So I will just show you, not to bore you too much with it, but still to show some interesting observations. So first of all, we had 218 applications, which was a lot because we didn't even expect to get so many. I believe around 30% were international and 70% Ukrainians. So, but applications were from all over the world. So Ukrainians who were living abroad as well, but also in Ukraine. Some interesting stats on how did you learn about the school. So when we started advertising it, I think the major contributor to spreading the knowledge was actually colleagues, teachers, and friends. So you should thank your great friends, teachers, and colleagues because they spread the information about the event. And so because the questionnaire actually allowed multiple choice answers, so I think Facebook as a social media played one of key roles to spread the word in general. And so maybe some fun stats. Like this is how raw data looks like. So I had to curate it a little bit so that it's a bit, there are fewer non-uniform answers. But basically, Twitter was not useful at all. And this is probably consistent with the observation that most of scientific community left Twitter completely in light of recent events. And I definitely recommend to create accounts or if you don't have any in this social media, because some of the scientific knowledge keeps somehow being discussed in Blue Sky. Well, Mastodon is sort of dead. I think at least part of network I'm in there, so not a lot is happening there. But I would say a lot of scientists also escaped into LinkedIn. So people advertise papers, conferences, and everything basically there now. And then another highlight I wanted to make, I don't know if this has been discussed, where to search for grants, jobs, etc. But I think for evolutionary biology community, especially important, I believe my pointer, sorry, didn't, but evolutionary directory, which is for short EvolDir. Not a lot of people seen the info there, but this is who in the end came to the school, I mean. But this is actually quite a big repository of advertising of anything new happening in evolutionary biology field. So this is a website. It looks like it has a design being from 90s, which is, correct me if I'm wrong, I think that's when it actually started to be on. So basically, yeah, you'll be able to take the link later because this will be, I guess, will be part of the directory we have for the school, this presentation, I mean, so you can later look it up. And so basically it has different subdirectories for each of the interests you might have. You can also email the curator of the school and they will actually advertise your own event. So if you want it to reach like really broad audience and it's somewhat related to evolutionary biology, this is really a place to go. And so, for example, our school also has been advertised here. So that's how some number of people actually learned about the school specifically from here. But for international scale, definitely recommend to monitor and maybe you will find some internship opportunities, other conferences, other events that are maybe less advertised on places like LinkedIn or Nature Jobs or anything else. So to go back to our statistics, so roughly 75% of people wanted to participate online and 25 in person, which is a good ratio and gives us estimate really how many people we could expect both in person and online for also future events. We had quite even distribution between people who have bachelor, master, and PhD degrees, and also significant fraction of people without degrees. And this, I don't know if that's maybe just only alumni from high school or who are doing undergraduate degree, or maybe even high school students participated at some point, or at least intended to, and I don't know if in the end decided to participate. Yeah, so regarding workshops, so we covered three major, most wished groups of workshop, which was bioinformatics, grant writing, and what's the other one? Motivation letters. Unfortunately for entrepreneurship, yeah, schedule was already very busy. It was difficult to find someone quickly. And then, well, question you all wondered about, you've done great at it, answering what is evolution. So this great job here. This was mostly for fun and to understand if people don't want to actually just to sabotage the event, but well, everyone was behaved well. Yeah, so some brief summary. So we had 26 speakers, and which means we had 26 lectures, and then we had also so we had 10 workshop lecturers, and which were split into 7 workshops, which was great. We really were excited about the enthusiasm of the organizers and of the speakers whom we were inviting, and people would suggest what they can offer, and that would be really great, really, really great. I wanna take in a few minutes more here. I really would like to highlight Denis and Rob, who actually reached us out and wanted to say that they wanna give a talk or organize a workshop. And this is really great. I think that kept us also being even more excited about school. So thank you for joining in the event, and for Denis especially coming in person as well. This is really great. Then this is, I'm abusing my organizer power. So Sebastian, who gave talk today, he actually wanted to advertise that he's looking for master student, PhD students and postdocs, and there are possibilities to work remotely. Please email him. And I do think the work will be somewhat related to what he spoke about today. So if you are interested in microbiome and evolution of microbiome, please get in touch with him. Then, and finally, this is one of last slides, I think. So what was the attendance of the school really? So pink circles are basically total number of views, which counts in-person attendance, number of people in Zoom, present on average across most of the talks. And so YouTube views is basically what people watched already after streaming was ended. So yeah, so live streaming wasn't very actively happening. So maybe 1, 2, 3 people were sometimes there, sometimes 7 even, I think, one of the days. And I'm sure one of those people was me, because I was often checking in if there are no questions there or anything technical. People say there is a problem or something. Well, in person, it's the same number of people, which I'm not sure now, like what exactly was final number, but it's more than 30 people. And yeah, so YouTube views, they will pick up probably when people catch up. The main idea behind having a stream and also the posting videos on the YouTube is that we actually can, like people in different time zones can actually catch up later and watch. So Monday has a lot of views already, which is more than 200. But of course, next days were more recently, so it will pick up eventually. And I also quickly put numbers just now, an hour ago, for Friday. So of course it will grow eventually, but majority of speakers agreed for their lectures to be public. So they are there. If you want to remember something, you are interested about something, you can always go back and look at the website, YouTube channel of Karazin Kharkiv National University. And I think it's biological sciences YouTube channel. So yeah, within the university YouTube channel. So yeah, I think that's about it regarding stats. And well, it looks like you've had a lot of fun there, attending also social events. And yeah, it's been a pleasure also to be part of organizing committee. I've learned a lot myself, and I'm looking forward hopefully some days to meet you in person as well. And yeah, so what's next? This is not a promise yet, but of course we're thinking how it's gonna grow and what we're gonna do. So we generally intend to continue. It can depend, of course, from funding and other circumstances. But overall, the idea is to continue with this. So we're looking forward for your engagement. Please spread the word among your friends. And yeah, I think this is more or less it. I think one important announcement, which still maybe in-person organizers will do. So we will have several surveys that we will need you to fill in, so we can actually, for next events like this, we can improve it. And we also would like to have some summary, like I showed some slides on how many views it had, etc. We want to sort of create a resource for people to learn how to organize event as well. So with this, thank you for your attention.
Thank you, Alexander. Do you have some questions in the audience? Maybe you know what about geography as for online participants, countries, etc.
Yeah, I should have probably put slide on country-wide distribution. Problem is that we didn't initially expect a lot of international audience. So there are quite a lot of people from Europe, but also we've seen participants from India, Pakistan, China, Turkey. I think couple of participants from USA, from France, from Germany, from Netherlands. So yeah, at least couple of people from maybe 20 to 30 countries, basically. And that's roughly corresponds to 30% of people who are international, something like that, I would say. I would need to dig into the table itself, and then we can actually tell more specifically um, from where. But yeah, there were quite a lot of people from everywhere. And so what I know also, this is in person, some people ask. So a lot of people were interested in 3, 4 talks, for example, and were not necessarily interested in the entire event. So this is a power of online or hybrid events where you can really pick what you would like to listen to. Because the idea was for in-person audience, yeah, you commit to the event, you listen to all the talks to broaden the knowledge and everything. But for a lot of people, they want to learn the updates of those specific topics maybe, and for them that was really great opportunity. So I think people from broad audience, from all whatever 200 people we got, I think everyone had slightly different goals, and this is really great. So we kind of adapted for everyone uh, to achieve what they want. So if you wanted to listen just for one talk, that's fine. You just listen to the talk. So yeah.
Thank you.
Maybe we can get some feedback from the audience who are here. Maybe somebody want to take a word about. We have volunteers.
Yeah, I want to express my sheer overwhelmedment by this event because, first of all, you collected like one of the maybe the most prominent scientists, the most interesting works, the most interesting lectures, just in 4 days, like 26 speakers in 5 days. It's very well. It's a lot of information, but I want to say that, of course, we are diverse. We evolve differently, like in biology. Some of people are microbiologists, some are zoologists. I'm a physiologist. Some are geneticists. But I think that such events, they really broaden our minds. They really broaden our knowledge. And well, for example, I couldn't even imagine that it's possible to combine so diverse, so different parts of materials in one brain. And I want to say that such difference really helps to understand the whole situation, the whole subject, like deepen my knowledge. Because I think I had like 3 or 4 courses of evolution through my lifetime. I I think in these 5 days, I got more knowledge than in previous like 3 semesters. So I'm really thank you. Thank you, Danny, for coming. It's very touching. Thank you very much. I really hope that we will meet in year or two, because such events must exist in Ukraine. I'm so happy and I'm very touched that we have such an opportunity. Thank you.
Thank you very much. We have one more volunteer.
I would like to say for how grateful we are for the organizers, for the organization in general, how good it was, how it was good with timing and for great conditions of living and great conditions of watching the information, getting it, and for the questions and opportunity of reflections of this. Thank you so much. You're great.
Thank you very much. One more.
Okay, so first of all, I'll be very short, just not to take our time, but I think that events like that, they were useful for our career orientation, because here we can meet a lot of like people with whom I'm definitely sure we will work together in future, and we can make some network connections that will basically boost our careers. So I strongly, I really want to express my big gratitude to the people who hold this event. So thank you so much. We really appreciate it, and all the knowledge is appreciated. So thank you so much, guys.
Maybe we have some feedback from our online participants. Guys, you are welcome to take a word. Yeah, we have some. Please. Valery?
Can you hear me?
Yeah, we can see you.
Yeah, I see people waving. I want to express my sheer and sincere gratitude. It was awesome. Like I took vacation on my work to participate in it, and it was awesome. Thank you for technical part, because sometimes on such events, online part is could be buggy and so on. This time it was awesome. Thank you for organizers, for professors, and for actually everyone who made this possible. Thank you for all the people who gathered to learn and share knowledge. So it was awesome experience, even online. So I actually envy a bit to everyone who participated in person. So thank you.
Thank you very much. We have some also very nice feedback from Angelica Kobrin and from Sergei Litvinov in Zoom chat, and Tatiana Alexeyeva. A lot of greetings, a lot of thanks.
You can read, I think. You can read.
I can read, okay. I can read. Many thanks to the organizers and lecturers on behalf of the staff of the Institute of Cell Biology and Genetic Engineering of the National Academy of Science of Ukraine. Deep greetings to the organizers and speakers of this wonderful school for an incredibly productive time, broadening our horizons and enjoyment of pure science. A special thanks for excellent organization. Дякуємо за старання. Дуже приємно було слухати цікаві лекції. Всі доповідачі старалися. Гарних нам всім творчих ідей. Так, ну, Валерій виступав. Yeah, thanks for making it easier for students to learn about modern science from Valeria Vassilieva. That's probably it for now. Yeah, that's it for now. Our friends from Czechia, would you like to make some feedback, please?
Yeah, thank you very much, all of you. It was a great experience. We are so sorry because we just got a call that our bus got cancelled and we have to catch a bus in an hour. So we need to leave like in a rush. But thank you very much for this great opportunity. We learned a lot. And yeah, we keep fingers crossed for your country. Yeah.
Thank you for coming and for interest for such event.
Okay. So maybe some people from organizing Svetlana. Ярослава Степанівна.
Do you want to? I think from behalf of organizing committee, I want to say thank you, all of you, for involvement of the event, for your great interest, for your great questions, for really good discussions, and for all what happened this week. And we hope that you learned a lot, but it looks like this. And also, I hope that you have new ideas to your research, to your work. And after this, it would be a good movement for you in direction of science to do nice and great things. At least some of you would be evolutionary biologists and who can continue this work which was started with Dobzhansky. So I think, do you want
from the side of the host from Uzhgorod National University, I would like to express my thanks for all participants and for all speakers. You're welcome in Uzhgorod in next time, and I hope that we will produce more ideas, not only in evolutionary biology, in different directions of modern science. And we would like to meet you in Uzhgorod. Приїжджайте, ми будемо вам завжди раді. Спасибі вам велике за участь. Дякую.
Я лише доповнюю, тому що багато сьогодні було сказано, що, знаєте, Ужгород – це таке місто, яке є на такому перехресті культур, історій, і місто, яке завжди готове до нових знайомств і співпраці. Тому подальшої нам співпраці, вам успіхів великих, тому що ви наше молоде покоління і на вас дуже велике сподівання. А без науки немає нічого насправді. Тому що я завжди кажу, що ті країни багаті, де наука на високому рівні. Не ті, які багаті там покладами, а ті, які мають дуже високий рівень наукових досягнень. Тому я вам бажаю дуже багато всього, але в першу чергу, звичайно, нам закінчення війни, перемоги нашій країні, і щоб ваше покоління вже працювало і творило у мирній Україні, незалежній і, я би сказала, найкращій країні світу.
Зараз я Сашу попрошу. А виведіть Сашу нам ще на екран, будь ласка. Олександра Майстренко. Щоб він теж був. А ви
Ну да, ви кажіть.
Okay. I would like to say thank you for everyone from also from our Karazin National Kharkiv University. I hope nearest time we will be able to gather all together in our university. And I wish you not only to be fruitful in science, but to get some kind of satisfaction from the job that you do. Yeah, not only being busy, but also being involved, being happy, being really satisfied with your activities.
Тож, а ви щось будете казати?
Бачите, дуже радий, що стільки людей приєдналось. Да. Сподіваюсь побачити теж особисто більше і так. Я не планував нічого більше говорити, але ну. Дякую всім.
Alexander Maestrenko, our financial director. Welcome.
So I
So-
think that USEP 2025 is officially closed.
Yeah.
Yeah.
Пару організаційних моментів. Да. Будь ласка, не забудьте заповнити анкету. Це перше. По-друге, буде там в тій теці, яка за QR-кодом, буде підтека, куди ви можете скласти свої білети, щоб вам зробили рефінансування, ті, хто по гранту тут. І фотки білетів. Да? І ще такий момент, для того, щоб вам зробили рефінансування, треба реквізити. Будь ласка, надішліть їх на пошту ЮСЕБ собака, то, що звідки вам там, не собака, равлик мене завжди поправляють, Karazin.ua, да? Бо реквізити, в теку якось складати не комільфо. Yeah. So, да, сертифікати також ми вам електронні розішлемо, да? Розішлемо. Ні, мабуть, в теку викладемо, там нічого такого особистого немає. Тобто кожен собі завантажить потім. Я нічого не забула? Може, запитання?
Це все. Але сертифікати тільки після заповнення анкети.
Ні, нам насправді дуже потрібні ті фідбеки для того, щоб здати звіт для спонсорів. Дякую всім. Велика вдячність Вікентію, який провів з нами тут цей тиждень, замість готуватись до перескладання екзаменів.
І також Сергію, який все це зробив дуже магічно у Зумі. Але він знаходиться у Харкові, він Зум робив нам з Харкова.
Да, доброго дня.
Доброго дня.